blood grouping has different theoretical aspects, will be discussed in the hematology theory classes. For the practical part, just you know need the initial knowledge about the blood groups, blood types, and as you determine the blood groups, very simple experiment. For the examination purpose, when you have to face the hematology practical exam in this physiology first phase university exam, whenever you perform an experiment, the examiner should or may ask you questions related to its theoretical aspects. You will have to show your experiment results, and then you will ask about the theoretical aspects. In this session, in this particular session, we will not discuss about the theoretical aspects. There is a we will take it in a separate lecture session. Just you need to know the preliminary things, I think, which you already know from your school level, that blood groups, it was blood grouping. This in 1900, one person called Landsteiner described this, the present, at least the ABO blood grouping. There are number of antigens, number of different antigens on the surface of RBCs in the human blood. The ABO system is the one which is the major one which is followed, and which is participating these antigens and antibodies participating in the agglutination reaction, which is of primary importance due to the consequences. There are various types of mismatching of blood during blood transfusion from one person to another, one blood type to another. If it is transfused, there may be various agents. The most important which we are to consider is for the ABO system. What it says, Carl Landsteiner said the laws of the two simple laws, which will seem simple now, that will, if one antigen is present on the surface of the RBCs in the blood of one person, one particular individual, that individual's plasma would not contain the corresponding antibodies. So antigen which is designated as capital A, the corresponding antibody is denoted by the Greek letter alpha. So if A is present, alpha won't be present. This is what the Landsteiner's first law says. Sometimes this is, say, if you even in the theory exam, if you get just what are the Landsteiner's laws, or if you are asked to state the Landsteiner's laws. So if one antigen is present, where the antigen is present on the surface of the RBCs, antibodies present in the plasma. So if one particular individual, this A antigen is present, the corresponding antibody won't be present. This is the law. The second law is just the opposite, and it is very simple. If one antibody is present, then the corresponding antigen won't be present. That means if, say, alpha is present, then A won't be present. If beta is present, B won't be present. So this is the first law, and this is the second law. What follows is that if A and B are both present, none of alpha and beta can be present in the plasma. If none of the antigens are present, then both the antibodies will be present. So A, B are the antigens which are recognized in the ABO system, alpha, beta are the corresponding antibodies respectively. If one is present, then if the antigen is present, corresponding antibody won't be present. If the antibody is present, corresponding antigen won't be present. And sometimes the second law, after if we go by the first law, the second law may it will feel it is a bit weird. It is obvious actually. So if one is present, otherwise not be present. And according to this blood grouping, we usually say, just remember this, we usually very commonly what we say, what is your blood group? Say somebody saying my blood group is A, then we are coming to positive or negative. Blood group is A, blood group is B, B positive, A negative. What is your blood group? But actually blood group, this is what we commonly say. Actually blood group is the whole system, ABO system of blood grouping. What we commonly say as blood group, that is actually blood type. That means very strictly speaking, we should say my blood type is A positive, not my blood group. Blood group is taking into consideration the whole system. So, blood group system is the ABO system together, but one particular individual's designation, that is called the blood type. What we commonly say, what is your blood group, but it should be called what is your blood type or what is my blood type. That is A positive. Positive and negative is determined by the this Rh antigen. Rh antigen again present on the surface of RBCs. What is and this was discovered a little later. In 1900, ABO system was classified. This Rh system came into existence or first described in 1940, okay, by the same person, Carl Landsteiner, and there another person whose name I just can't recall right now. Carl Landsteiner, another person together described this. Okay, I can't recall at this moment. There were two persons. ABO system was identified only by Carl Landsteiner himself, and in 1940, 40 years later, the Rh system was described. So, and that Rh antigen is Rh, this Rh term comes from the term, it was first described in, that means this was derived from a species of monkey called the Rhesus monkey, and it is, the antigen is labeled as capital D. Like in ABO system, it is like A and B, here the antigen is labeled as capital D. Now, as in the ABO system, there are naturally occurring antibodies in the plasma, namely alpha and beta. They may or may not be present according to the blood type, but there are naturally occurring antigens occur. But in case of antigen D, there are no naturally occurring antibodies. Okay. In case of antigen A and B, there are corresponding naturally occurring antibodies, which if one antigen is present, corresponding antibody won't be present in that individual's blood. But in case of Rh antigen, there is actually no naturally occurring antibody. There is nothing, if it comes to our mind that if corresponding of A or alpha, B is beta, then corresponding of D should be delta, but actually there is no existence of delta. Okay. So, if antigens A is present in one person's blood, then what would be the corresponding antibody won't be present. Which antibody is present in that person's plasma? Blood means A will be present on the surface of the RBC. So, what will be present in plasma? Anti-B, or which is designated by beta. So, now antigen D may or may not be present. So, if it is present, this blood group, strictly we should say blood type is A positive. If A present, D is not present, beta is present, it is A negative. And it is similar for the others. If B is present, D is present, alpha is present, it is B positive. If B is present, D is not present, and if B is present, alpha will be present, then it is B negative. Right, this is simple, very simple. Without the presence of D Rh antigen, it is negative. With the presence of Rh antigen, it is positive, and the rest will be with the, according to the presence of A or B antigen and alpha and beta antibody, and if A is present, alpha can't be present, if B is present, beta can't be present. Likewise, if A and B are present, then antibodies, none of them will be present. And along with, if D is present, it is AB positive, blood type is AB positive. AB present, so alpha beta won't be present, and D is not present, then AB negative. if none of A and B are present, that means A not present, B not present, alpha and beta are present, D is present, then it is O positive. If no antigen is there, alpha beta are there, it's O negative. Now these 8 blood types are possible in the AB, considering the ABO and Rh systems together. So how many blood types are possible? 8 blood types. The experiment, simple experiment in the lab is to identify which one of these 8 a particular individual is having in the blood, that is which of the blood types among A positive or A positive negative, B positive negative, AB positive negative, O positive negative, among these 8 which is the individual is possessing. So any one of the 8 is possible, not more than one. So for that what we do, various authors are writing just simple differences. We do it over here like this. We take a slide, okay, we take a slide and divide it into 3 parts and label it as A, B and D, okay. Whether we will just see whether agglutination reaction is occurring in something called the agglutination reaction, just will explain that, A, B and D, whether it is occurring in these areas. We may, for beginners we may do it as, we can do it on separate slides, I am telling why, but this is sufficient. So these are, what are these called? These are called antisera, antiserum plural is antisera. So this is labeled as anti-A, this means that these have particular color, anti-A is blue colored, anti-B is this yellow colored, anti-D is this grey colored. These are made so that they react with the corresponding agglutinogen. Antigens in blood, these antigens are called agglutinogens and antibodies are called agglutinins because they participate in a agglutination reaction. Agglutination is a clumping reaction. Agglutination is, if we just go by the literal meaning of it, it is clumping. So this agglutination reaction occurs in the body when it, naturally in the human body when it may occur, if what the Landsteiner's law says, alpha can't be present if A is present, beta can't be present if B is present, if the corresponding anti-agglutinogen and on the agglutinogen, antigen or agglutinin antibody comes together, then agglutination reaction occurs. So if A and alpha come together in the same individual, agglutination reaction inside the body, okay. So here we are making the agglutination happen so that we can determine the blood group. We are in the anti-A, antiserum, this is called antiserum. These are made because how these are made? By injecting naturally occurring agglutinogens in the body of an experimental animal, say rabbit, an experimental animal, so that antibodies are produced and then that serum is drawn, that is called antisera, and this contains the antibodies. So if antibodies A are antigens A are made to produce antibodies corresponding to alpha or similar to alpha, then that will be present in the, anti-A antibody, antisera. So if this is mixed with the individual's blood, whose blood type is unknown at this point of time, at one point of time I want to determine it, then if the antigen present on the surface of the RBC in that individual is A, then agglutination reaction will be observed. It can usually be observed with the naked eye. If the naked eye it is not clear, we see it under the microscope, okay. So for that, so you can obviously understand if the blood taken on the slide should not be this antisera and the blood, the different types of antisera must not get mixed, then the obviously results can't be obtained. So as beginners, sometimes what we sometimes do is that we take 3 or 4 different slides, at least 3 different slides for the A, B and D, but actually it is not required. If we carefully do it, we can do it in the 3 sections of the same slide, just by taking care that these do not get mixed, because we will take just a small drop of antisera and we will take a small drop of blood, which we take by, if we take capillary blood, by pricking our own finger. Just I will add the blood to the antisera, okay, and mix it, and after mixing it, waiting for 10 minutes, agglutination reaction will occur if the corresponding antigen is present. So, if antigen A is present in a in an individual's blood, this mixing it with antisera will show agglutination reaction. So, on one section of the slide labeled as A, labeled as capital A, I mean label it is anything 1, 2, 3. If I just remember that 1 is for A, 2 is for B and 3 is for D. Just labeling it, if agglutination occurs in this A section, that means the antigen A is present on the surface of the, we can conclude antigen A is present. We do not know what is present, but if agglutination is seen with this thing, then antigen A is present. If agglutination is seen with this thing, then antigen B is present. If agglutination is seen in this, naturally occurring antibodies do not occur to antigen D, but we have prepared it by injecting where there is no D. If antigen D enters a person, if any animal in a human being enters a person, corresponding antibodies will be produced. Naturally occurring antibodies are not there. That means by nature there is nothing as such, but if D antigen is introduced in a person in whom antigen D is not present, then corresponding antibodies will be formed by immunological reactions. So, this is that antisera. So, if agglutination occurs with this, then we conclude that antigen D is present in that blood. So, now the results are simple. If agglutination occurs in all the 3, then the person's blood group is, it contains A, B, D all antigens. So, the person's blood group is concluded as AB negative. If it occurs in A and D part but not the B part, the person's blood group is A positive. If it occurs in B and D part but not A part, it occurs in the person's blood group is B positive. The person's blood means the blood sample we are taking is B positive blood type. If there is no agglutination in the D part, then it is negative, and according to A or B or both, it is A negative or B negative or AB negative. So, very simple experiment. So, it will require, but after the agglutination requires few minutes, so we usually see the result, observe the result after 10 minutes. After 10 minutes, whatever the 8 to 10 minutes, whatever the result is, we conclude that from the examination point of view, after you complete the experiment, it is a very short experiment. So, from the examination point of view, I tell you this, if this experiment is there, then there will be other experiments also. If till now in whatever is in your syllabus, we have discussed differential leukocyte count, hemoglobin, and today we are discussing blood groups. So, if blood group is there, there will be another maybe 1 or 2 more small experiments. If differential leukocyte count is there, then 45 minutes to 1 hour you get for the hematology exam. One single experiment, and whatever the marks allotted will be for that single experiment. If blood group is there, along with there will be hemoglobin, or there are other topics to discuss that will be given along with, because these requires maximum 10 minutes, but the exam time allotted is 45 minutes to 1 hour. So, there may be more experiments together. It is better to, in a simple experiment, there is not much credit for the experiment itself. So, there will be theoretical questions along with. So, if there are 2 or 3 experiments, I think in my opinion it is more scoring from the exam point of view, and this is a very simple one. So, after this, whatever result you get, you have to report to the examiner, and you will be asked how what is the procedure, how do you do it, and the relevant theoretical questions. So, determining this is very simple. We will do it in the lab. We will take the antisera, we will add the blood to it and just determine. You can this is so simple that even in this particular setting you can determine your own blood group. If sometimes it is not possible, at least like this, for beginners to identify what is agglutination, the second slide we take, we take normal saline, that is control solution. Okay, the normal saline is called control solution, that means test solution. Adding blood to a test normal saline, obviously normal saline does not contain any antiserum and antibodies, so there will be no agglutination reaction. Just to understand the agglutination reaction, what it looks like after 10 minutes, obviously every person's blood will show some agglutination reaction. It cannot be that if agglutination reaction is not there in any of the A, B, D portions, then it is O negative. So we can differentiate it or compare it by adding blood to normal saline, where there will be obviously no agglutination reaction, but usually it is so a very distinct reaction, so it can be seen easily. If it is not possible, we see it under the microscope. Now, if it is not possible to see with the naked eye, which is very rare, then it is better that we take 3 different slides for the 3 different antisera and note the 3 different these 3 slides one after another under the microscope, and maybe a 4th slide for this control solution, so that under the microscope it is very clear. So this is very simple, we will just do it in the lab. It will take maximum 30 minutes, there is enough time. Okay, so do it, and this thing I think if you do once, you don't need much practice. This is a very simple thing, you just add antisera and blood and see the agglutination reaction. Okay, so only a single class is allotted, even a single class, a whole 2-hour class is not required for this. So this is a very simple experiment, but remember, from the exam point of view, in the examination, if you get this one, then you will get an additional experiment. So let's proceed to the lab. Just he mentioned, then I remembered that never write it as plus or minus. This is a very common mistake. One of the students just mentioned plus minus. So what may happen when you will become professionals, using a sign like minus, just think it in your head. A minus sign may be easily converted to a plus sign. So this blood groups, due to blood groups, many legal complications occur due to wrong blood grouping. So always write, make it a practice right from your this first year of medical school, that you will write as A positive, the whole word positive and negative, so that there is no chance of tampering. Did you listen? I thought I will mention it in the class. Never write as plus or minus. That is a common practice. Minus may be easily tampered. Just by giving another, just a vertical stroke, it may be converted to plus. Write blood groups as positive and negative whenever you write any document. At least, even if you write in short, sometimes it is better to write the whole thing always. Even if you write it short, POS stands for positive, NEG stands for negative, but don't use the hyphen sign as minus and this plus sign as plus. Any document can be tampered. You as a medical professional sign a document, and that may be tampered, and in case of blood grouping, you will face legal consequences. So this is A positive. This is with the same blood, so it is A positive. So 2 or 3 blood samples are there. That's why I prefer taking capillary blood, and it will help you with pricking also. It has to be free flowing. Without that, you can't do experiments. In the usually it is the question is given in the exam that determine your own blood group. So you have to prick your finger, and the experiment procedure is whatever you did now. You prick your finger and just mix the antisera and the blood and see the agglutination reaction. So did you notice what is this? A and A blood group, A, and this is agglutination occurred or not? Occurred. So it is A positive, A negative. This blood sample, that is not called coagulation. This is another common mistake. This is called agglutination. Coagulation is a same completely separate thing. Don't utter coagulation along with blood group. Coagulation is blood clotting. This is not blood clotting. This is agglutination, clumping reaction. This is the same blood?
Yes, sir.
Okay, A positive. Topics. What topics have we done? Those who have joined newly don't know. We'll try to arrange some revision classes. So topics in order will be study of the compound microscope. If you forget, you write down study of the compound. There is nothing to forget. Those who have performed study of the compound microscope, followed by preparation and staining of a peripheral blood smear. That was the second topic. Second topic means topic is same, DLC. If you see your syllabus, topic is DLC, but to arrive to do this experiment, we must learn to stain. So first class was on study of the compound microscope, which we mention in the practical notebook. Then preparation and staining of a peripheral blood smear. Just listen. Then it will be identification of blood cells or identification of components of blood under the microscope. This is the third one. Fourth one you write is as differential leukocyte count. Then estimation of hemoglobin and then determination of blood groups. Give the dates. You are in batch A. So experiment dates, we are some classes we have repeated, but experiment dates you give as, you see the calendar, whenever you have joined, after that give a date, either a Tuesday or a Friday, it will suffice for one experiment. Don't give other days. You are group A, so Tuesday or Friday, and it should be a working day. Now as far as I remember, there was no holidays except academic holidays except the Sundays. So you give any Tuesday or Friday, but do it in order. These 6, whatever mentioned now, do it in order. This compound microscope, study of the compound microscope, then it will be preparation and staining of peripheral blood smear, then identification of blood cells, then differential leukocyte count, estimation of hemoglobin and blood grouping. So those who have joined late, so 6 classes days you have to give at least 6 days. So have you got 6 days, 6 Tuesdays and Fridays after joining? So as it is a hematology practical notebook, these are official documents. If you have not got, then you may give one Wednesday or Thursday, but better you keep it on Tuesdays and Fridays for group A. When your groups will become permanent, then according to roll numbers, then you give days as such. So you may give other days, but as you have to maintain it, any working day. But remember, these have to be in order. Dates will have to be in order. Okay. Don't give any date before your date of joining, as this is an official document. So maybe they give a date before, but you don't give, but you just mention all the dates after you have joined. If somebody has not got 6 Tuesdays and Fridays till now, then they may give another date. So that is still manageable. So you did not attend classes on this identification of cells, staining? No. Hemoglobin did you attend? Hemoglobin was done just last week, no? Hemoglobin, write that date, that class was officially conducted. Now if you have these reasons, you have to then we'll try to revise the classes, but if you have some reason not to attend the class, write an application to the HOD, Physiology, and you have, I think you have documents with the student section also.